In the central nervous system, noradrenaline originates in several discrete areas, especially the locus ceruleus. These areas send noradrenergic neurons throughout the brain that exert widespread effects. Noradrenaline is synthesized from the amino acid tyrosine in a sequence of enzymatic reactions culminating in the conversion of dopamine to noradrenaline in synaptic vesicles. Once activated by an electrical impulse traveling down the presynaptic neuron, these vesicles fuse with the membrane, releasing noradrenaline into the synaptic cleft. Two main types of adrenoceptor are found on the postsynaptic neuron: alpha adrenoceptors and beta adrenoceptors, both of which have several subtypes. The beta-1 subtype is the key receptor on the postsynaptic adrenergic neuron. Beta-1 receptors are linked to stimulatory G proteins and adenyl cyclase. Activation of beta-1 receptors leads to an increase in intracellular levels of cyclic AMP. The presynaptic neuron also has both types of adrenoceptor, although the key receptor is the alpha-2 subtype. As with beta-1 receptors, alpha-2 receptors are linked to G proteins. In contrast with beta-1 receptors, however, the G proteins linked to alpha-2 transmission are inhibitory, and activation of the receptor leads to a decline in cyclic AMP levels. Activation of alpha-2 receptors by noradrenaline switches on a negative feedback loop, causing a dramatic reduction in any subsequent release of noradrenaline. In this way, the neuron auto-regulates the release of its neurotransmitter. Beta-2 receptors, also located on the presynaptic neuron, are activated by low levels of noradrenaline in the synaptic cleft. This positive feedback loop increases release of noradrenaline when appropriate. Noradrenaline is removed from the synaptic cleft by a reuptake mechanism. Inside the presynaptic neuron, metabolism by monoamine oxidase on the outer membrane of mitochondria renders noradrenaline inactive. Any free noradrenaline remaining in the synaptic cleft is inactivated by catechol-O-methyl transferase, or COMT. Cell bodies of 5-HT neurons are found predominantly in the raphe nucleus and brainstem, but the major fibers innervate many regions of the brain, including the hippocampus and frontal cortex. 5-HT, also known as serotonin, is synthesized in the presynaptic neuron from tryptophan, an essential amino acid. After manufacture, 5-HT is packaged into synaptic vesicles found in the nerve terminals. When stimulated by an action potential, these vesicles fuse with the membrane, releasing 5-HT into the synaptic cleft. 5-HT1A receptors are most common in the hippocampus and raphe nucleus. 5-HT1A receptors are linked to the second messenger system, consisting of a G protein negatively coupled to adenyl cyclase. Activation of the receptors causes the G protein to directly influence the ion channels, increasing conductance of calcium and potassium. Consequently, levels of cyclic AMP decrease, while diacylglycerol and inositol triphosphate increase. Because of this inhibitory effect, the action potential along the postsynaptic neuron is considerably weaker than along the presynaptic neuron. 5-HT2A receptors are concentrated in the medial prefrontal cortex of the brain, a region thought to become abnormal in schizophrenia. As with 5-HT1A receptors, 5-HT2A receptors are linked to G proteins. In contrast with 5-HT1A receptors, however, The G proteins linked to 5-HT2A transmission are excitatory, and activation of the receptor stimulates phospholipase C, leading to the formation of diacylglycerol and inositol triphosphate. Phospholipase A2 is also activated, releasing arachidonic acid. The increased formation of DAG and IP3 causes the intracellular levels of calcium to rise, along with activation of protein kinase. Chloride and potassium travel through the membrane normally up to this point. Responding to the rise in calcium, calmodulin enters the postsynaptic neuron and begins to soak up the excess calcium. As a consequence, the efflux of potassium from the cell is slowed. Due to these stimulatory effects, the electrical impulse along the postsynaptic neuron is considerably stronger than in the presynaptic neuron. 5-HT is quickly removed from the synaptic cleft by a reuptake mechanism. Inside the presynaptic neuron, metabolism by monoamine oxidase on the outer membrane of mitochondria renders 5-HT inactive. A deficiency in 5-HT neurotransmission is thought to be the underlying cause of many mood disorders, such as depression, obsessive-compulsive disorder, and mania. Selective serotonin reuptake inhibitors, or SSRIs, block the sites where 5-HT is taken back into the presynaptic neuron. When 5-HT is released from the neuron by electrical stimulation, SSRIs stop the normal reuptake mechanism and prevent the deactivation of 5-HT. In this way, 5-HT neurotransmission is enhanced, correcting the deficiency common in many affective disorders.